We have covered the basics of DNA replication in the last video, so now let's dive a bit deeper into a few more details about how the process works. We know that DNA polymerase adds new nucleotides to an existing DNA strand in order to replicate it, but the direction and orientation of how this process works matters. Before we talk about that, we first need to understand that both DNA strands and nucleotides have terms that describe their orientation, called 5' and 3'. This naming refers to the carbon ends of each nucleotide. We call that within a nucleotide there is a central sugar that contains 5 carbons, called ribose. These carbons are numbered 1 through 5 starting at the rightmost carbon, which is 1, and then moving around the molecule clockwise up to carbon 5. You can see here that carbons 3 and 5 are oriented away from the nitrogen base and connect to each phosphate group to create the backbone of the DNA, which makes them great candidates to use when describing their orientation compared to other nucleotides within a complete DNA molecule. When we have a complete double-sided DNA molecule, we can see the 5' and 3' ends of each side of the molecule, which we call the 5' and 3' terminals. And because DNA strands are facing opposite directions, the 5' and 3' terminals are opposite as well. Now, why is this important for DNA replication? The new nucleotides that get added to each original strand during the process of DNA replication are always added in the 5' to 3' direction of that new strand by the DNA polymerase. This means, based on the orientation of this picture, that the new red strand will be added with DNA polymerase moving to the right because the top blue strand is oriented from 3 to 5. So the new red strand will be 5 to 3 going the opposite direction. This orientation is followed for the other original strand, meaning the DNA polymerase will move in the other direction as it builds the new strand from 5' to 3'. In order for DNA polymerase to attach to each original strand of DNA and begin the replication process, it requires a double-sided strand to adhere to. To accomplish this, an RNA primer is added to the DNA, which provides an anchor point for the DNA polymerase. Once attached, the DNA polymerase can begin the replication process. But because of the 5' to 3' orientation of the newly added strand, and the DNA continuously being unzipped by the helicase, there are parts where new pieces of DNA are added continuously and other parts where they are added in fragments. If the 5' to 3' end of the new strand is moving towards the direction that the DNA is being unzipped by the helicase, the process of adding the bases is continuous and only requires one RNA primer to start. As the DNA unzips, the same polymerase molecule can just keep moving in that direction, adding new nucleotides. But at the same position on the other original strand, the nucleotides cannot be added continuously because it is being unzipped in an opposite direction that the polymerase has to add the bases, which we know is always 5' to 3' for the new strand. In this scenario, the new DNA bases are added in fragments, which requires a new RNA primer each time to anchor the polymerase and is later connected into one seamless piece. These fragments are called Okazaki fragments, named after the married couple who discovered them, Riji and Senetko Okazaki. The RNA primers are later removed and DNA bases are added in to fill in the final gaps. More on that on the next slide. At this point, we have the process of DNA covered in terms of a general overview, but there are a few other definitions of enzymes within the process you need to know for the AHL exam. These include being able to understand and define the terms DNA primase, DNA polymerase I, DNA polymerase III, and DNA ligase, all within a prokaryotic cell system. Let's go over each one individually in order of how they are used in DNA replication. DNA primase is an enzyme that assembles the RNA primer we discussed on the last slide, and that RNA primer is used as an anchor point for the DNA polymerase to attach to. We have not discussed this yet, but there are a few different types of DNA polymerase enzymes found in both prokaryotic and eukaryotic cells. DNA polymerase III is the main enzyme used in replication, which adds new nucleotides to the existing strand. It also has the power to proofread the DNA and make corrections if incorrect bases are added, which we will discuss on the next slide. DNA polymerase I has the ability to add and remove nucleotides by both breaking and linking the bonds between them. This polymerase is used mainly on the lagging strand between RNA primers and Okazaki fragments. It attaches to the space between the fragment and the primer and moves in the 5' to 3' direction, removing the RNA primer nucleotides and replacing them with DNA nucleotides. But because DNA polymerase I, and all DNA polymerases for that matter, can only attach nucleotides in the 5' to 3' direction, it leaves a gap between the previous fragment and the newly placed DNA strand. This is where DNA ligase comes in. This enzyme connects any remaining phosphate sugar bonds between nucleotides within the lagging strand. And when all is said and done, 2 complete DNA molecules are created with the help of these enzymes. The process of DNA replication is a very exact one, but not due to a lack of molecular effort. It has built-in proofreading mechanisms to ensure the smallest amount of mistakes possible, with the obvious goal of 0 mistakes. In bacteria, all 3 DNA polymerases, 1, 2, and 3, have the ability to proofread using 3' to 5' exonuclease activity, which means they can remove or cut out the nucleotide in that direction. For example, when an incorrect base pair is recognized during replication, DNA polymerase III reverses its direction by 1 base pair of DNA and removes the mismatched base. The DNA polymerase reads the newly added base before adding the next one so that it can be corrected. If the newly added base has paired correctly with the base in the template strand and all is fixed, the DNA polymerase continues with the replication process.